Hello, everyone. Thank you for tuning in to In Plain English, the podcast that's all about making science approachable, open source, and jargon-free. I'm your host, Jamie Moffa, and this is the 5th installation of Interviews in Pain Research, a series of interviews that I conducted at the United States Association for the Study of Pain conference last April in Seattle. But before we get to the episode, I have a quick announcement. Last Wednesday, I recorded the very first In Plain English live show in Philadelphia, Pennsylvania at the University of Pennsylvania, co-hosted with the Community for Rigor. It was an absolute blast, and I had such a good time. I'll be releasing a few clips on Instagram from that event in the coming weeks, and then the full episode will be available on the podcast feed on Tuesday, July 2nd, as well as a video feed available on YouTube. So stay tuned for that. It's going to be amazing, and to all those of you who showed up, thank you so much. So without further ado, let's get to the episode. Our first interview for this episode features Aleyah Goins, a graduate student at the University of New Mexico. Her research focuses on developing a new model of chronic back pain in mice that is less invasive than previous models. And she and her lab hope to use this model to develop new drugs for chronic back pain and understand more about the role of the immune system in perpetuating chronic back pain.
And can you start by introducing yourself?
Yeah, my name is Aleyah Goins. I go to the University of New Mexico, Department of Anesthesiology. My mentor is Dr. Sasha Alice, and he studies chronic pain, and more particularly ion channel and neuronal excitability. And so we hope to do drug discovery and figure out ways to treat chronic pain. So today I am going to be presenting about a chronic back pain model. And so back pain is a little bit controversial in a mouse model.
Right.
Because they walk on 4 legs. Yeah, exactly. So no, we don't make our mice walk on 2 legs. But I use an injection with a drug called urokinase type plasminogen activator. And for short, I just call it urokinase.
Okay. What does it do?
And so and anytime you get like a cut, your blood starts to clot, and so that's called coagulation. And this drug actually prevents coagulation.
Okay.
So it keeps things flowing.
Okay.
So whenever I inject this drug into their spinal ligaments, it prevents clots from forming from the injection.
Okay.
And so what that does is actually increases immune cell migration, or increases the amount of immune cells that can move into the spinal ligament.
Okay.
And so what we're seeing is that these mice, whenever they have immune cell moved into their spinal ligaments, it's another way to say they have increased inflammation around their spinal column.
Right, and then that leads to back pain.
That leads to back pain.
Okay.
Yeah, so we have pain behaviors. The mice have increased mechanical sensitivity, they have cold sensitivity, and they have gait disturbances. So I actually just noticed this, that they walk in their cages, and that's not super extreme. It's pretty subtle, but they kind of just waddle just a little bit more than their counterparts. And so I ended up using a gait test, and I found that the angle of their paws is a little bit outturned more than their controls.
Okay.
And so that's pretty consistent with patient symptoms, that patients will have gait disturbances, cold sensitivities, and mechanical sensitivities. And that's how they experience their pain modalities. And then we also did this thing called flow cytometry, which is a way to measure how many immune cells are infiltrating into the dorsal root ganglia, or the nerve bundle that's near the spinal ligaments. And so we saw that there was increased amounts of immune cells, increased inflammation, and that is also correlating with those pain behaviors.
Yeah, that's really cool.
Yeah.
And then I also see down here you have some data on the excitability of neurons from these animals.
So
or
actually,
is this different?
No, you're that's pretty close. So actually, this is a naive mouse, which means that it's a mouse that hasn't been touched. So we took the dorsal root ganglion neurons from that mouse.
Yep.
And we applied urokinase directly to the neuron.
Okay,
And
gotcha.
so the reason for that is because I wanted to show our hypotheses, which is that immune cells are responsible for the pain behaviors, not necessarily the drug itself.
Okay.
So this experiment is sort of like a control to make sure that the drug is not causing the changes in their pain behaviors.
Gotcha. So if you just like plop the drug down onto these sensory neurons that don't have any immune cells around them, then nothing happens.
Nothing happens, yeah.
Okay, so...
Yeah. And then, and again, I use human dorsal root ganglion neurons, and then we saw the same effect, but except I used a readout of ATF3, which is a proxy for cell stress. So cells only release it if they're stressed. And I saw no significant differences between 3 conditions with urokinase or with their controls. So that's basically saying that urokinase is not harmful to cells. It's actually just an endogenous protein. It does its job. It regulates fibrin clots by affecting the blood coagulation, which is a normal process in pretty much all biological animals.
Right.
And then, but if there is a presence of an injury, such as an injection into their spinal ligaments, then we can see those increased amounts of immune cells, aka increased inflammation, which can lead to these pain behaviors.
Right. And so how will this model then help test, are you thinking it'll help test drugs related to pain or like back pain mechanisms?
Yeah. So there's my goals with this project are twofold. So one, the previous ways to induce back pain in mice were pretty invasive. It involved constriction of their spinal cord or degenerating their disc or causing massive damage to the muscles or ligaments surrounding it.
Right.
But with this model, it's like one injection, they don't feel it, they don't react, we're done.
Right.
And so so that's one way. One way I want to contribute to science is by just providing a new tool that's a little bit nicer to the mice, and that's easier and faster to induce.
Nicer to the researcher too.
Yeah, it's not. Yeah, I am personally invested that I don't have to perform surgery for 10 hours straight.
Right, massive spinal surgery, no fun.
Yeah, and so. And then the other side of this is that this is a way to induce inflammation directly around the spinal ligaments, and I think that that's closer to a patient experience of back pain, where they might have one day where they might bend over wrong or pick up something wrong. You sprain that ligament, you get an inflammatory response, and then you kind of have that persisting pain over a couple of weeks. And so being able to look at the neuroimmune mechanisms involved in that, because of that one incident of inflammation, we might be able to discover which immune cell types are best suited to be treated as a drug target for future drugs.
Gotcha. Yeah, that's really cool. Thank you for going through your poster.
No, thank you so much.
Our second interview features Aish, a postdoctoral fellow at Ted Price's lab at UT Dallas. Her research focuses on examining the sensory neurons in the dorsal root ganglia, or DRG, from human organ donor patients, either those who had diabetic neuropathy or controls that did not have diabetic neuropathy. And in her research, she was examining the individual cells and neurons within these sensory neuron bundles to see whether there were any changes in between diabetic neuropathy patients and controls.
And can you start by introducing yourself?
Hi, my name is Ishwarya. I go by Aish. I'm from Ted Price's lab at UT Dallas. So today I'm going to be talking to you about what are the different characteristic cell types that are present in the diabetic neuropathy in the DRG. It's not been shown before, and this is the first time we're trying to characterize them. And so just to give you a brief overview, diabetic peripheral neuropathy, or DPN, affects about 50% of the adults that have complications with diabetes. Most often what they undergo is foot ulcers or even lower limb amputation. So it's kind of what's happening is what we need to know and understand. If there is a difference in cell type proportions, is things changing between certain cell types, if there's something going around with the neurons itself, as well as if there's any neuromodulatory interaction between the immune cells or non-neuronal cells and the neurons. And that would help us develop better therapeutics, especially for the DPN population itself.
Right.
So what we did is to do single nuclei sequencing, where we took organ donors who have a pain history of diabetes or control in the sense which no known pain
Right.
in there. And what we did is single nucleus sequencing, where we actually took the DRGs from them, and then we used this kit called the 10x Flex Kit, which allows you to fix the nuclei. So, and then go through the downstream processing. Just by fixing, it allows the integrity of the nucleus to be intact.
Yeah.
And then you follow through the single cell sequencing pipeline.
Okay, and just for, because this is more of like a general audience podcast, so all of these are like human sensory neurons that are coming from these organ donors.
Correct.
And so then you're fixing them with like formalin or paraformaldehyde.
Yes, it's kind of like a proprietary fix that comes with the 10x platform.
Right.
But yes, you do fix with formalin.
Yep, and then sequencing each individual cell.
Yes, and that's why it's called single nuclei. We basically take out the cell membrane out of it and just think about just the nucleus. And so what we actually see over here is that there are 7 distinct cell types that are present in the human DRGs, irrespective of whether they are the control, which is like the no pain, or the DPN. All populations are represented in both of them. And you see 7 different clusters, where SNAP25 and perforin are the genes to mark for neurons itself, which is like over here that I marked in blue. CD14, CD163 are the classical markers for macrophage. And so that's what I kind of call them as immune cells.
Right.
There's a cluster down there. COL-A1 is a classical marker for fibroblast. And so that's what we identify as a fibroblast cluster out there. FAP-P7 is another classical marker for satellite glial cells. And so that's what you see around this portion. And then MPZ and MPB are those classical markers for Schwann cells. And so that's how we kind of identify the cluster population based on the marker genes that are available. We see a small cluster of adipocytes as well as endothelial cells out here. So
there's actually quite a lot of different kinds of cells in the DRG. It's not just the neurons. There's supportive cells, and there's immune cells, and there's fat cells. That's really cool.
Yes, and so what you have to actually see over here is that between the control and the DPN, DRGs, you see like a different proportion of each cell types that are present over here. So some may be increased, some may be decreased. And so our idea is to understand that what are the different genes and markers that are there, and what is that is decreased or increased in this whole thing. So what we're trying to do is, I guess for the pain forks, mostly everyone is mostly interested in like the neuronal clusters, and is there a change between the control and the DPN data set? So 2 distinct clusters that I want to bring up is one is this cluster number 2, which is kind of missing in the DPN donor. So this cluster actually represents the parvalbumin, which are the big diameter A fiber nociceptors. So we think that either the neurons are dying or there's kind of like a reduction in its expression.
Yeah, in the patients that have the pain.
Yes, in the patients who have diabetic peripheral neuropathy. And then the second one that I want to emphasize is cluster number 7, which is kind of reduced as well between the control and the control. There's an increase, and then the diabetic, there's a decrease.
Right.
And if you look at the marker genes that are available, you see that the NPPB or the IL-31 RA, which are kind of like the itch nociceptors that are present on the skin. So if you look at the symptoms of diabetes, sometimes they have like nerve dieback, or they have like tingling sensation and everything. So it's possible that these innervation from the skin is actually represented in the DRGs itself. It needs a little bit further validation for us to be 100%, but this is something kind of cool to know, and maybe we could develop therapeutics targeting these populations itself. Another one, a lot of talk has gone about macrophages in this symposium itself. And what you can see, there's a lot of changes between the control and the DPN dataset. So just to give you a certain clusters, one is cluster number 12, which we think are the oxidative stress pathways that are upregulated, which would kind of make more sense because when they have inflammation or diabetes, they're kind of like increasing their ROS production and everything. And another one is the cluster number 2 and 3, which are kind of reduced over here. What we think, what we look back is to see that these two clusters have the adaptive immune response. So in the DPN data set, what we notice is that possibly the adaptive immune response is not functioning very well. So there could be some sort of like a hindrance or some sort of immune neuromodulation that is missing, which we can actually try to help feedback. And lastly, what we have to know is SPP1 and OSM are kind of known as the inflammation macrophages, which is increased in the diabetes data set, or people who have diabetic neuropathy. So these are like some really cool findings that we've found in the macrophage population.
Right. So they have more inflammation, but like less of this adaptive immune response, and more of these like this was like the oxidative stress. Okay.
Yes.
Yeah.
And so another thing that we notice is in the immune cell clusters, we see an increase in T cells, especially the T inflammatory phenotype, which is actually represented by this gene called TBX21, which is a transcription factor for the Th1 response. Th1 response are those genes who release pro-inflammatory cytokines, and that seems to be increased in the DPN donor data set.
So again, there's this like really pro-inflammatory state
Correct.
going on.
And lastly, what I wanted to just show is there's a complete increase in the adipocyte population compared to the control in the DPN dataset. We don't know why this could be an increase, but what we noticed that one of the adipokines is called the adiponectin, which is highly upregulated in the DPN dataset. This needs a little bit more validation and characteristic, but this is one of the first time where we have characterized DRG neurons with diabetic neuropathy.
Yeah. This was really, really cool work. Thank you for walking through that.
Thank you very much.
Our final interview for this episode features Chen, an assistant professor at Ohio State University. Her research focuses on patients with facial pain and trying to understand the changes that patients with facial pain undergo, specifically with respect to their sympathetic nervous system, which is the part of your nervous system that sort of governs fight or flight responses.
So can you start by introducing yourself?
Hi. My name is Chen, and I'm assistant professor at Ohio State University College of Dentistry. I'm a facial pain specialist, and my research interest is in stress, physiology, and facial pain. And
so can you take me through your poster?
So sure. In this is a pilot feasibility study. So we're just trying to look at how in a human physiology lab to set up some stress tests and then to continually monitor people's cardiovascular response and recovery from those stress challenges in the lab. So we're just testing out different activities in lab and to see how people react to those. And because there are some mechanisms that indicate that autonomic nerve function is associated with how they feel pain.
Yeah. So because this is a podcast for a general audience, can you kind of describe what an autonomic nerve is like?
The autonomic nerve usually it's there are two sets. There are two branches of autonomic nerve. One is called sympathetic.
Yep.
That's your fight and flight nerve. That when you're facing a dangerous situation, this nerve really active, and then you just stop thinking and either run away from the predators, things like that.
Yeah.
Your heartbeat beats up, your blood pressure goes up, so make sure you can run, survive.
Yes.
Your parasympathetic nerve is on the other hand, it's the balance at the opposite side. It makes you relaxed, digest, reproduce. All of these activities are related with parasympathetic nerve function. And literature find that in chronic pain, facial pain patients, the function of sympathetic and parasympathetic nerve are changed. For example, one of the biomarker for parasympathetic nerve function is called heart rate variability, and that has shown reduced in chronic pain patients.
Oh, interesting.
Yep. And from this study, what you can see here is heart rate.
Yeah.
Like when you're stressed out, your heart rate goes up, right?
Yeah.
And in this study, we compared. This is a pilot study, so we only have four patients and two controls. So the controls means they have no health issue and they're young. Controls age between 18 to 35.
Yeah.
And then the cases are those people who have some systemic issue like sleep issues, psychological issue, body pain, plus they have facial pain.
Yeah.
And those, and you can see there, when they react and recover from challenges, different types of challenges in the lab, their heart rate goes up and down in the controls, which is the blue bar. You can see the heart rate goes up when they facing a challenge. And the second one is when they recover from that challenge, the heart rate goes down.
Right.
So the sympathetic and parasympathetic nerve regulates pretty well. This is another hand gripping exercise. That's really your sympathetic nerve active. So you see when they're doing this hand gripping exercise, their heart rate goes up. This blue bar represents the change. The bigger the change, you see the taller the bar. So, and then when they after their stress challenge, the recover phase, their heart rate goes down a lot.
Yeah.
So you can see this variation in the controls very well, but in the cases like the orange bar, those are the facial pain cases.
Yeah.
That doesn't change too much, so it's blunted. So there's some dysregulation in that patient group, and that's something we need more study on.
Is that just because they start out with a higher heart rate, or?
No, actually, if you look at the baseline, the blue bar, that's the control group. They actually have a little bit higher heart rate, and we don't know why that is.
Interesting.
And then this is interesting too. It's called pre-ejection period. That's cardiac impedance. That's a marker reflect sympathetic activity. So the taller the bar, that means you have longer pre-ejection period. That means you're more relaxed.
This is the time when the ventricle of the heart is filling up with blood before it contracts.
This is the period between the nerve tells the heart to pump out the blood to the time the blood actually pump out from the blood from the heart. So that period. So the longer that period is, the more sort of relaxed, in plain language, more relaxed, less sympathetic tone. So, but you can see cross every, this is the baseline, and then cross every stage of the challenge and recovery, the controls have a longer, relatively longer pre-ejection period, so they're more relaxed. and the cases are relatively more sort of stressed out. That's
Okay.
And then, and why does that happen? We still need to do more study. But this is pilot, and our protocol went pretty well. Seems like everybody tolerate the protocol. It's pretty safe. Nobody say there's any adverse events. So we're hoping to increase sample size and see if we can really figure out what is going on there.
Right. Yeah, that was really interesting. Thank you so much.
Thank you.
Thanks everyone again for tuning into this episode of In Plain English. Once again, this episode has featured interviews from Aleyah Goins, Aish, and Chen. You can find their posters as well as previous interviews on the website at inplainenglishpod.org. Our logo is by the wonderful team at Biographics. That's Jessica Higginbotham, Tanya Lintz, and Yolanda Campos. Our music is by Sam Brunwasser. You can find more of his work on SoundCloud at Visuals Now Beats. And finally, be sure to follow us on Twitter, Facebook, and Instagram at Plain English Sci so that you never miss an episode or exciting news and updates about In Plain English. Again, that's PlainEnglishSci. Thanks once again for listening, and come back next time for another episode of In Plain English.